Hi, everyone. Welcome to Osteobites. Thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on Osteobites, we are joined by Dr. Brian Flesner, a veterinary oncologist at Maine Veterinary Medical Center. Thanks so much, Dr. Flesner, for joining us on Osteobites today. We are thrilled to have you. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Flesner after his presentation. A little bit more about our guest today. Dr. Flesner earned his Doctor of Veterinary Medicine with honors from the University of Illinois in 2010, and then he completed a rotating internship at the Veterinary Specialty Hospital in San Diego before pursuing a residency in oncology at the University of Missouri, where he simultaneously earned his master's degree and became a board-certified veterinary oncologist. Dr. Flesner has held faculty positions at Louisiana State University, the University of Missouri, and the University of Pennsylvania, where he directed oncology clinical trials. And he also served as the Director of Clinical Oncology at PetDx, focusing on cancer genomics, and he continues to contribute to clinical trials to advance veterinary oncology treatments. And beyond his professional endeavors, Dr. Flesner is an outdoor enthusiast who enjoys kayaking with his rescue dog, Brew, and hiking in the Ozarks. He's a passionate sports fan, following the Illinois Fighting Illini, Drake Bulldogs, Kansas City Chiefs, and St. Louis Cardinals. And when he's not in the clinic or in a kayak, you can find him working alongside his father to care for their beef cattle herd on his family farm. So definitely a busy guy. Just a few announcements before we get started. We hope you can join us at our annual Factor Osteosarcoma conference. The countdown has started for real. I think we're about 2 months out. Our conference is June 26th to 28th in Salt Lake City, and our scientific program is available. I will put a link in the chat where you can find it. We will have 7 scientific panels, as well as our very popular patient panel. And I did want to highlight 2 scientific breakout sessions, which are great opportunities to engage in the conversation. We will be offering a young investigator mentoring and coaching session in partnership with SARC, which will be led by Dr. Scott Acuno from Mayo Clinic, Dr. Lara Davis from OHSU, and Dr. Kevin Jones from the University of Utah. And then the second breakout session is an open forum on advancing single-cell research into clinical care for osteosarcoma patients, which will be led by the Cure OS working group. Participants will review evolving recommendations on using single-cell technologies to study intratumoral heterogeneity and the tumor microenvironment, and discuss strategies for translating these insights into personalized treatments. And comparative oncology is infused into this discussion, as we have DVMs, PhDs, and MDs working together on this. So that group is going to be led by Ivana Papasic and Corey Weistick from Ramona Sloan Kettering Cancer Center, Anand Patel from St. Jude, Emanuela Palmerini from University of Miami, and also Dan Regan from Colorado State University. So I'll put a link to find more information on Factor and how to register in the chat. We do also have a conference room block at the Grand America Hotel in Salt Lake City, where the conference is taking place. So you can take advantage of a discounted room rate by booking at the link, which I'll put in the chat. And then whenever we have an episode featuring work on canine osteosarcoma, I'd like to highlight our canine clinical trials resource on our website. It provides information and resources on participating in canine trials, as well as a directory of veterinary oncology programs and open canine osteosarcoma trials, including the study that Dr. Flesner will be discussing today. We also do have a comparative oncology playlist on our MIB Agents YouTube channel, and I'll put a link into that if you want to catch up on past episodes. And lastly, I just wanted to plug a cool event that's happening in early June. Comparative Oncology is being featured in a PBS documentary series called Shelter Me. The episode is called The Cancer Pioneers, and there is an in-person premiere in Bryn Mawr, Pennsylvania on June 2nd. The film is about groundbreaking clinical trials for dogs with cancer and how these treatments are being applied to people with cancer. And the film features some of the leading scientists at the NCI, the University of Pennsylvania, the University of Illinois, and the University of Wisconsin. And there will be a panel discussion and Q&A following the film. So I don't think the RSVP form has been launched yet, but save the date. And if you are interested in attending, feel free to email me. I'll put that in the chat, and we can connect you with the event organizers. All right. So I think we are ready, Dr. Flesner, to get started with your preso.
Thanks for having me. It's been a bit since I've done Osteobites. I did it, I think, season 1, which was a while ago, but when I was at the University of Missouri. And so this is kind of fun to to be back. So thanks for having me and inviting me. We're going to talk about a new clinical trial that's led by Ardan Animal Health for canine osteo, and it's through the USDA. So we'll just go through a little bit of background and then talk about the canine study itself. So hopefully I can advance. Hopefully that's showing okay as well. My conflicts of interest, I'm a paid project manager for the Ardan clinical trial. So I am evaluating the data, overseeing data collection, trying to make sure the trial runs smoothly and follows USDA guidelines. But I'm not an employee of the company or have stock. I have another conflict of interest, especially for Anne, if she's on here or if she's going to listen in later. I'm a part-time farmer, which is kind of funny. When I was younger, I was like, I'm definitely not going to be a farmer. And then life has led me to have to help to take care of the family farm. So I took the picture on the bottom left for Anne, last Osteobites. She asked me a question about where John Deere built the first tractor, or where John Deere, the company, was founded, because my family drives John Deere tractors. And it is in Illinois. It was funny. The clip, she made like an edit that looks very Star Wars for like the, if you have time, go back in time. It's really funny at the very end of the talk. But anyway, there's me in Illinois, where John Deere was founded as a company and the first tractor was built with a John Deere. So kind of funny. We also have beef cattle. So we just finished calving season. That's one of our little calves on the ground.
That might be the cutest cow I've ever seen.
It's a pretty cute, and I think we called that one Randy, which you're not supposed to name them because then you get attached. But I think with my family we call that one Randy. So, okay, outline for today. We're going to talk a little bit about cancer and the immune system. I think many others have discussed this, and so I'm just going to go over kind of some things I think that tie cancer together with the immune system and how to think about it. I'm going to focus in today on antigen-presenting cells and T cells, specifically cytotoxic T cells. And then we'll really talk about immune checkpoints. Sadly, I know immune checkpoints haven't been as groundbreaking in osteosarcoma, or really but just not that beneficial in human osteosarcoma. We haven't really studied the main molecules much in those tumor types in vet med. We really looked at them in mast cell disease and melanoma. And so I'm going to talk a little bit about checkpoints and then really go into like veterinary approaches, what we have commercially available, and then I think where we're hopefully going with checkpoints in vet med. And then we'll talk a little bit about this study and how it's not a true checkpoint inhibitor, but it is involved in the immune checkpoint blockade. So we'll talk about the background of Ardan's study scheme, and then the outcome measures we're following, and then just a little bit about study sites. And thankfully, MIB has already shared this on there, as Christina just mentioned, on the list of cancer clinical trials, veterinary cancer clinical trials that are open. And then I'll also show where it's available on the AVMA website. So the hallmarks of cancer, when I was a younger training oncologist, we started with, I think there were 6. And then Hannah Weinberg upped it to 10. And now this latest one, which was in 2022, I don't know, I'm starting to run out of space on the circle to remember, but I think we're upwards of 12 or 14. Back in earlier teens, they did update with a couple more immuno hallmarks of cancer. And so cancer has to gain a lot of things to be able to live and thrive in the body. And a couple of those really focus on the immune system. And one of the first things it has to be able to do is to avoid immune destruction. And so that's really important. And the second is that there are good types of inflammation, bad types, and lots of gray types, all kinds of gray zone in the middle. And so there is chronic inflammation and other pro-inflammatory scenarios where actually you can promote tumors. And so there's this balance of getting the immune system to recognize and kill cancer, but not have bad inflammation, if you will, that can actually promote cancer. And so there's two sides of the coin there of almost good and bad inflammation. And so anyway, just to remind everybody, those are two of the pillars, if you will, of the hallmarks of cancer. Cancer is a genetic disease. And I just go back to this to remind everybody of how does the immune system really think with cancer cells and real cells in your body. And so this schematic on the left is actually from my time at PetDx. I didn't go into detail, but I was at Penn, and then my father got pretty sick back in 2022. And so I had to pivot pretty quickly to go back and forth to the farm from Philly And then I've been kind of on this consulting locum and relief work journey for the past few years. And so this was when I worked with PetDx. They were really excited in performing liquid biopsy in pets and had some really neat stuff with osteosarcoma. I presented at a factor, I want to say maybe 22 or 3, or maybe 23. And so this schematic is just a reminder of the genome itself. And when we really think about cells and chromosomes, and osteosarcoma has one of the most aberrant and wild chromosomal and genetic signatures, if you will, and it's varied between patient types. But you have to go all the way down to, hopefully I can get my cursor to show, to the individual base pairs themselves in the DNA. And when we really think about those base pairs, the whole goal is for that to be transcribed to RNA and then eventually translated into proteins. And so we think a lot about genes and other things, but then what the immune system is going to worry about is actual proteins. And so whether it's exogenous factors, and I just had a talk with a family today about their dog with squamous cell carcinoma in the mouth. And they said, how does this happen? And I said, well, in people, we do stupid things to ourselves often, and smoking or drinking or chewing tobacco. And, we made the joke of, I doubt your dog was a chain smoker, but, there are things that people do, and maybe all of us are exposed to in life that can cause cancer. The other side of things is that there's endogenous factors that promoting inflammation. If you have chronic inflammation in your body, if you have other immunologic abnormalities that could lead to cancer, you have both the exogenous and endogenous factors. The accumulation of that DNA damage does over time result in usually abnormal proteins, and those abnormal proteins help cancers grow and spread, and so we get that cancer formation. And so I took these images from a few different places to think about where our genome lives in our cells, how that's then affected by our environment or our own bodies. It does then have to turn, those DNA molecules have to eventually turn into proteins. And protein is then actually how our immune system usually recognizes and kills infected cells or neoplastic cells. And so the different types of antigens, which is then a kind of a breakdown product usually of proteins, can be self-antigens. So, it's something that you have in your own body that's mutated. I think a lot of us think about what mutations are present in osteo, which is all kinds, oftentimes. Do you have overexpressed self-antigens? And so in human breast cancer, we think about overexpression of HER2. And for osteosarcoma patients and doctors, we really think about HER2 a lot now after the listeria study that we did in dogs and now as in clinical trials in people. There are specific genetic products that are unique to cancer that can make oncogene products. And then I do also then talk about, viral antigens. And so in some cancer types, not osteosarcoma, but some cancer types, viruses themselves can be the antigen, whether that's their DNA or their RNA, or they're making a protein in the cell itself. And so there's some specific lymphomas and other things where the viruses can be involved too. And so your immune system's job is really to survey the body for abnormal proteins or abnormal antigens, if you will, that way upstream come from, your own genes themselves. Sometimes exogenous things like maybe a virus. So how do you surveil and survey the body? How does this work? And so we really split the immune system into the innate immune system and the adaptive immune system. The innate immune system, I think of always as the first line defense. These are your friends that are always around. And I think a lot of cancer patients always think about your PMNs, your neutrophils. In veterinary medicine, we always talk about nukes. And so those are your first line of defense against infections. And so we think about those oftentimes when we're getting chemotherapy, but they're really important in just natural life. And so the innate immune cell I'm going to really focus on today is the dendritic cell. And those oftentimes live in your skin or they're very close to skin tissues. And their job is to either eat things that are infected or just to kind of taste, if you will, things that are around, and they chew it up and they display those antigens, those broken down pieces of protein or other molecules as antigens. They're often called antigen presenting cells. Their main job is to talk to cytotoxic T cells. They also can talk to some T helper cells. And so those are either CD8 or CD4 respectively. And so the job is to say, hey, this is what's in this environment. Let me show it to the T cells that actually are then the workers that are going to say, hey, we'll go either kill something that's infected or tell the rest of our immune system to pay attention. And so this is just to show the main two cells I'm going to focus in on. Innate immune system, this is stuff that's always around, always actively surveying your body, and then the adaptive immune system, which can then pivot and make decisions based on what the discussion they had with antigen-presenting cells or dendritic cells. And the reason I'm focusing in on those two is really because of the clinical trial we're going to talk about. So when I talk about those two, they actually have to communicate directly. And I took a certification course a few years back from the Society for the Immunotherapy of Cancer. And so some of these diagrams are from that training course. And so just to know, I did not make these myself. These are from immunologists and oncologists that worked with that course. I really like the way they described immunologic synapses. We often think of synapses as like nerves that are going to talk to each other and send messages, but your immune system kind of does the same thing. These cells don't just run by each other and say, tag, you're it. They have to communicate with each other. And that communication usually we think about as 3 steps. And so those 3 steps are here. So I'm not going to quiz anybody on immunology after this. But the first one really always is a T cell receptor. And so that's on the green cell. That's going to be, if my arrow's not being very friendly today. Can you see that, Christina? Is that showing up,
yes,
the arrow?
I see your cursor. Okay. Yep.
So the blue receptor here on this green T cell is a T cell receptor, and its job is always to match up, depending on what type of T cell it is, with an MHC receptor on an antigen-presenting cell or another cell in your body. And so in this case, I think the diagram has MHC class 1, and so that's on every cell in your body, but it also is on these antigen-presenting cells. And so that MHC class 1 receptor on this dendritic cell here on the left is going to communicate with the T cell receptor on this cytotoxic T cell. In this case, if it's going to communicate with one, it's a CD8. And those two things say, hey, we have a match. So the antigen-presenting cell or the dendritic cell is going to chew up things in its environment or taste, if you will, and it's going to display that little red bubble on its MHC class 1 receptor. The T cell then says, okay, my T cell receptor matches. So whatever that little piece of protein or molecule is, the two have to have a match to lock, if you will. And so you might say, oh, great, that's it. Those things then communicate and we move on. But it's not as simple as that. You have to have other factors involved with that communication. And so, if we really said, let's just focus in on MHC1 and TCRs or T cell receptors, things would be a lot easier to get the immune system to work. But you have to have these cofactors. So the second thing that's shown here is number 2 is a yellow and kind of a navy receptor. And those are co-stimulatory molecules. And the main ones we talk about are CD28 and CD80. And so I think you can see here CD28 being on a T cell, CD80. Immunology, when I was in vet school a long time ago, was always alphabet soup to me. There was all these names and numbers, or letters and numbers, I should say. And I was like, I don't know if this is going to be important for me. And it turns out as an oncologist, yes, very much so, it is important. So I guess for everybody here is really you need the first lock between a T cell receptor and an MHC molecule, and then you need co-stimulation with different signals. And there's all kinds of letters and numbers, but those two receptors have to then also talk to each other. And that's actually then also where checkpoint inhibitors can come into play. And so this schematic is a couple images put together, but on the top you have stimulatory signals in the green box, and on the bottom is red, and those are inhibitory signals. And so the drawing just shows one set of signals touching each other and talking, but really it depends on who is touching and talking to each other, whether it's a go signal for the cell or it's a stop signal. So again, the first thing is you have a little antigen that's chewed up, that's from the environment, whether it's from a cell or an infection. That T cell and the antigen presenting cell lock, and they say, hey, this is a match, we should think about it. The second things that happen is either stimulation with a co-stimulatory signal or inhibition from like a checkpoint inhibitor. And I think everybody's probably aware now from drugs they've heard about or received of PD-1, PD-L1, CTLA-4, LAG-3. Those are all those co-stimulatory signals that are actually then going to be inhibitory. And so those things tell us either, hey, immune system, go kill stuff, or hey, take a back seat and just take a break. The third thing that has to happen is then you have to have further excitement to get other T cells involved, because one can't do it on its own. And so excitation with the rest of the immune system happens often with cytokines. And so we need really good T cell cytokines like interleukin-2. And so if anybody's had or thought of interleukins therapy that they've received, that's kind of the third thing here. And so you have an initial lock between T cell receptors and MHC. Second, either stimulation or inhibitory signals from other cell surface receptors. And third, excitation, hopefully, with cytokines, but you also can have inhibitory ones as well. Any questions? I know that that's a lot to digest in like a minute or two, but that's where a lot of this comes from, is that immunologic synapse communication between your innate and adaptive immune systems. Good?
Yeah, great, helpful illustration too.
Yeah, thanks. That was a good course from that organization. So the next step is, the slide is from actually a cancer.gov NCI diagram. And so I think Therese Winslow drew this one. I really like it because it just, it makes things a little bit prettier, and you see that locking kind of aspect. And so now, instead of a T cell and an antigen presenting cell, you have to think about T cells, then we'll talk to those antigen presenting cells, and then they'll go and look for similar proteins on tumor cells. So if the antigen presenting cell chewed up what was around it and said, ooh, there's cancer around here, you should go look for it, it's going to find that also on these cancer cells. And so the same receptor, that MHC1 receptor, is going to be on that tumor cell. That T cell receptor is still going to talk to it. This is where the stimulation or inhibition comes into play, is in the tumor cell could say, ooh, I'll put PD-L1 on my surface. The T cell will go in and say, I have PD-1. If we match, I stop and I don't kill. And so that's the checkpoint. That's the immune checkpoint. And so if that's what I always taught, is a don't eat me signal. And so the cancer cell says, hey, we have a match. That's cool, but you really don't need to kill me because I don't want to be killed. And so they put these immune checkpoints, this don't eat me signal on their surface. So instead of a co-stimulatory signal, you get a stop sign almost. And then the T cell will go about its way looking for something else to kill that it has a match with. So when you add in an immune checkpoint inhibitor, like I have up here Keytruda or pembrolizumab and some other PD-1 or PD-L1 inhibitors, those actually block those two signals from matching. And if so, then the T cell can say, actually, I can kill this cell because there's no immune checkpoint. And so it's like a negative of a negative, but the way to think about immune checkpoint inhibitors is they block the don't eat me signal. And so a T cell can then say, hey, I don't like this, I'm going to go in and kill. And so then you can see on the right-hand side of this graph, this image, actually then tumor cell death. So that is a little bit of a really basic kind of black and white of immune checkpoints and immune checkpoint inhibitors. And there's all kinds of examples. This is PD-1 and PD-L1, but that goes for CTLA-4, LAG-3, and then we'll show some other examples from other studies. So I'm here to talk about kind of a checkpoint inhibitor, but a different set of molecules. And this is one that I had heard of, but not really thought a lot about, just because there's not much scientifically reported about it in cancers that I've looked at, or really in osteosarcoma is my main study, or my main focus of research. And there's just not really anything about it. So CD200 used to be called OX2. I think sometimes still people will kind of use the same terminology between the two. That's receptors, they get all kinds of names. So CD200 or OX2. CD200 is a highly conserved glycoprotein on usually dendritic cells and then some B and T cells, but you can see it also on all kinds of endothelial cells or epithelial cells in your body. And then you could also say it's probably going to be on, eventually probably some cancer cells are going to take advantage of that if they come from those cells. CD200 has a CD200 receptor, and so those two proteins actually interact. And the downstream effects of this is that actually when those two bind, the B and T cells usually don't have as much cytokine production. And I remember that third step, and to really get a good immune response between cells, is for cytokine production to ramp up. I said interleukin-2 was one of the big things. But you need initial docking. You need to get either a pro-stimulatory or a inhibitory signal, if you will, and then cytokine production. And so the binding of these two proteins together reduces the amount of good cytokines that are in the environment. And so there's not as much excitement from the immune system. People will talk about tolerance or exhaustion, but really the B and T cells that are going to interact with that CD200 don't get excited, and they might move on down the road. So on this right-hand side of this slide, you can see a tumor cell that has CD200 that's binding a T cell, and then also to actually some really not as nice immune cells. And so here's an MDSC or myeloid-derived suppressor cell, or a tumor-associated macrophages. Those are actually the bad inflammatory cells we think of that suppress the immune system. And so there's actually interaction between those two that say, hey, let's actually prevent the immune systems from coming in. So it's kind of a two-pronged thing, is that either you can say, let's not make any cytokines to really excite the immune system. Let's actually bring in some immune suppressor cells to also suppress the immune system. So this pathway, it seems, especially in neuroendocrine tumors and some leukemias and melanoma, really suppresses the immune system from being active, which is not a great thing, especially for immunotherapies. Okay, so that's a little bit about CD200. It's not a new receptor, but I think it's a newer researched immune checkpoint pathway. This is a really nice schematic from Tim Phan's group. I know it's super small. I'm going to blow up each of the corners just to talk about those. We did a little bit of a summary about the immune system, how it works really, really briefly, and then what immune checkpoint looks like. So this I just want to talk about. I think you guys know what you have available in the human space. What do we have available in the veterinary space? So I'm going to really talk about what's commercially available, not what's experimentally available. So one of the big things that we've done, and I think you probably heard a couple of our veterinary researchers talk about, we have been doing CAR T-cell therapy. There's a couple groups out of the Midwest and also the East Coast that have been looking at CAR Ts, usually for lymphomas and leukemias. Some people have talked about different CARs on other types of lymphocytes for osteosarcoma. But CAR Ts are chimeric antigen receptors, so that's actually an engineered receptor to go after specific antigens or those little molecules that are presented on the front of cells. So CAR T-cell therapy is being studied in veterinary medicine, and there's several groups around the country that are looking to try to say, hey, can we get cures like are in people with lymphomas or leukemias with CAR T-cell therapy? And I know that people are really trying to figure it out for osteosarcoma specifically. The same thing we were talking about, you have to get T-cell receptors involved. You also have to get cytokine involvement. And so there's groups in veterinary medicine that have looked at intratumoral cytokines. And this specific one I'm showing is a melanoma study where they looked at anchored cytokines that stick into the tissue area. And so interleukin 2 and interleukin 12 are some of those examples. Cancer vaccines, that's a component of the study. And I'm going to talk about cancer vaccines have been used for a long time. You can go way back to the early days of tumor immunology, and that's where all of this started. They really don't work on their own. And so usually you need something with them to get some gusto, if you will, in your immune response. But cancer vaccines are an option. Usually you take a tumor and then you try to make antigens specifically to excite the immune system from that, add stuff to kind of, I always say, piss off the immune system. And then you give that, hopefully then to get, again, a T-cell response. And then the fourth thing really is then immune checkpoint inhibition. Much further along in human medicine, we're catching up in vet med, and I'm going to talk about a couple specific examples of immune checkpoint, well, really one example of immune checkpoint inhibition that we have available to us in veterinary oncology. My first really commercial experience with immunotherapy in veterinary medicine goes back to 2010. And that's when we actually got a vaccine, if you will, which was a tumor antigen called Oncept. I don't know if anybody's heard of that. It's kind of old news, if you will, in veterinary medicine. But this was our first commercially available immunotherapeutic. This was fully approved in 2010. It's owned now by, oh gosh, I need to make sure I say this right. I actually think it's Boehringer Ingelheim has it now. But the mechanism of action, this is what's crazy. And I just bring it up because I don't get to talk to I don't say humans very often, but yeah, human scientists are people that study people. So this is actually a xenogenetic vaccine. It's human tyrosinase. And so we actually give dogs human DNA, which is kind of mind-boggling, if you will. People have asked me about the flip side, and I don't know the ins and outs of it, but I think this was developed at Memorial Sloan Kettering in New York City with Bill Bergman, who is at the Animal Medical Center in New York. And they really looked at human, mouse, and dog tyrosinases. And eventually they focused in on using human DNA to make an immune response for dogs. The initial study, you can see the Kaplan-Meier down here on the right-hand side. The vaccinates lived a lot longer than the dogs that didn't get vaccine. And so these are dogs with melanoma that had surgery or radiation therapy, and or radiation therapy, and then got vaccinated. And it really seemed to, in this report, extend survival And then I have up in the corner actually the plasmid that's given, and actually then you can see the human tyrosinase gene that I give to dogs, which is kind of nuts. So anyway, this is part of my practice, standard practice for canine melanoma. And so it's immunotherapy that we give that's actually human in origin, if you will, with a lot of E. coli in there as well. But I just thought kind of neat to give you an example of immunotherapies that we use in the clinic commercially for dogs. The second one I really want to mention, and really this is what's commercially available right now, is Oncept and now Gilvetimab. And this is our first commercial checkpoint inhibitor. This is a canonized monoclonal antibody for PD-1. This is owned by Merck Animal Health. Its job is to, just like Pembro in people, is actually meant to block PD-1. And so I guess Keytruda is the branding that you would think of. It enhances interferon gamma secretion, which would be pro-inflammatory, and engages with PD-1 on CD4 and CD8 positive T cells. We don't really have a clinical trial available, but we have abstract presentations and some data that's kind of superficial from Morsi in 2021, and they looked at stage 1 through 3 mast cell tumors and stage 2 to 3 melanoma, and the certain dosage they gave. This was by itself, and so this is kind of exciting data when we think about these tumors with no other therapy, no steroids, no nothing else. They had an objective response rate of 46% in mast cells and 20% in melanoma. I think when you use that with other therapies like steroids for mast cells or like radiation therapy for melanoma, it really could go up. And so this is getting studied with surgery, with radiation, with other things a lot. Side effects were pretty low, but they did have 2 dogs that had severe anaphylaxis. And that's one of the risks with checkpoint inhibition is severe immune side effects, especially like allergic type reactions. And I think some of those studies that I've seen in human osteosarcoma, pneumonitis is a big thing. We think about like GI side effects from really inflammation in your gut or ulcers. And so, you really have to think about this could really excite the immune system, but you don't want it to go so far that it actually hits the rest of your body. So just to let everybody know, we also see those kind of side effects in canine patients that receive checkpoint inhibitors. This is pretty exciting. We haven't had anything for dogs really made. So this was the first one. You can see this is just a few years old. It's not fully approved yet. I'm hoping they keep doing the work. And we have some other checkpoint inhibitors in the wings, I think, that hopefully move the needle for us. So osteosarcoma standard of care. What do we do? I think you guys have heard this before, but I just want to mention what we do and then where does immunotherapy come into play for this. We do amputation. We don't do as many limb spares. We're really kind of stuck with only 2 locations, if you will, distal radius and distal tibia that can have limb spares in dogs. Dogs also don't like to use their legs that have prosthetics. They have 4, and so they often say, I don't need 4. I can easily get around on 3. And so they like to just oftentimes not use them or they just don't deal as well with limb spares. And so we really rely on amputation. We follow with adjuvant chemotherapy. We really don't do neoadjuvant chemo. Our chemo agents are platinums. The reason we don't do neoadjuvant is because ethically, we often think that dogs are in a lot of pain. Their tumors are usually late stage. And so we struggle to say, get some chemo, be painful, and have to wait for their amputation. So our go-to is amputation and then chemo. Amy LeBlanc and I think I saw her on the people attending. Hi, Amy, if you're here. I've mentioned her a few times. Let's do neoadjuvant. And so hopefully someday we start doing maybe something more similar to what's done in people. Speaking of Amy, the seminal paper, I think the highest level of evidence trial that we have was when we looked at standard of care in dogs versus standard of care plus rapamycin. It was a negative study, but it gave us a really good, true prospective, multi-institutional randomized study that said, what are we doing in dogs and what kind of survival are we getting? And so I think you can see historically with retrospective studies, our numbers look pretty good. And then when you actually look at standard of care in a prospective fashion, we're really getting in that group of dogs 9 months. And that's with amputation and chemo. Older studies said amputation alone is probably 3 or 4 months in dogs. It's really, really short. They get pulmonary meds just like in people without chemo. And so, we really need to move that needle is, do we do better with chemotherapy? Or I think what a lot of us are hoping is, can we add in immunotherapy? And I know that there's been talks about the Listeria vaccine and other types of immunotherapy to really improve that chemo response rate and duration. We're really working on that. Non-surgical options, if the dogs can't have a surgery, we think about radiation therapy, bisphosphonates, and pain meds. And then I think just popped in the Kaplan-Meier from Amy's study shows that there was really no difference in the groups of standard of care or standard of care plus rapamycin. So this table is actually a summary. Jeff Bryan at Missouri and Charlie Mites put together of different immunotherapies and where they've fallen. I took this and modified it and made it specific to osteosarcoma, and I focused in on disease-free interval. And so from Dr. LeBlanc's study that was multi-institutional around the country, the median disease-free interval, the dog in the middle of the road, how long it stayed in remission was 6 months. And so I think for us, that's a good idea of how long that what we do as standard of care keeps dogs in remission. On the human side, that would be terrible. You guys would laugh. You would say that's not enough remission. And so, yeah, we're trying to do better. There's a lot of numbers on here. I'm going to try to circle what's important. If you go back to, I think this is the 80s, I was like really, really young or maybe not born. Sorry to the Wisconsin folks. The initial studies looked at a bacterial cell component called LMTPPE, which is really mural tripeptide. That's actually like a component of a bacterial cell wall, like a mycobacterial cell wall. It really is like a non-specific immune stimulant. And so Greg McEwen's group looked at amputation with LMTPPE versus amputation alone with just placebo. And the dogs that got immunotherapy alone had a disease-free interval remission duration of 6 months versus 2 months if they just got placebo. But you can say if you just do an amputation alone, they really don't do well. They then added cisplatin to that immunotherapy, and really this is a rudimentary immunotherapeutic. And they got up to 11 months of remission, which is really good. You can see there's no other 11 on this slideshow. If you look at just cisplatin alone with placebo, it was 7 months. Sadly, LMTPPE isn't available in the States. I do believe it's actually, I don't want to speak on behalf of physicians, but I think it's used commonly. I'm not for sure of standard of care in osteosarcoma patients in Europe. I can't think of the name of what it's called brand name, but it's LMTPPE.
MEPAC.
Yeah, exactly. Yeah. I was like, we don't have it. We can't use it in vet med in the States. And so I'm not sure if our European counterparts can even get it, but used commonly in Europe. Yeah.
Yeah, and compassionate use available for humans, compassionate use in the US.
Oh, okay. Yeah. I have talked about it with residents during my career, and the data that is from dogs is pretty stunning. They looked at either sequential or concurrent. And I think they decided that concurrent is not useful. And so in dogs anyway, it was labeled, or not labeled, but thought to be used sequentially would be better. So you're not negating the immune response from chemo. But anyway, you have to fast forward quite a ways to see, did we do anything else? And so that was the late 80s, early 90s. You can go all the way then to, I think this is a late 20 teens or 2020s study out of Italy with Laura Marconato's group. They actually added salmonella in with chemotherapy, and they got a disease-free interval of 10 months. And so really the only double-digit numbers you'll see on this chart are dogs that got chemo and immunotherapy. If you look at then when we did our initial study that I talked about on the first Osteobites many years ago, the Elias clinical trial, we actually did immunotherapy alone, and that was adoptive T-cell with an autologous vaccine and then a cytokine boost. Our disease-free interval with immunotherapy alone, no chemo, was 7 months. So, I think there is something to say of immunotherapy. We have to figure out how does it work addition-wise with chemotherapy. So just some numbers to show you where we're at in the disease-free interval realm with dogs. So where does the current clinical trial science come from? Ardan is a company based out of the Louisville or Lexington, Kentucky region. They worked with researchers at the University of Kentucky, and they make a live cell vaccine that creates neoantigens after UV-radiated immunogenic cell death. Their primary adjuvant is CpG-ODN. It's an oligodeoxynucleotide. It mimics a bacterial adjuvant, kind of like that LMTPPE. It's not at all the same, but that's the goal, is to say, a non-specific immune stimulant. It's internalized by dendritic cells and has been shown to stimulate interleukin-2 and interferon gamma release. Those are some of our good cytokines. We want to perpetuate that immune response. They've vaccinated over 1,500 dogs with vaccine alone, and their internal data is shown here. These are survival times, not remission durations. And so they have some I think, kind of exciting, especially in hemangiosarcoma, data with immunotherapy alone. That's a really rough disease for us to treat. They have treated about 50 dogs with osteosarcoma, again, with vaccine alone, and have close to a 9-month median survival time, again, with just this autologous vaccine. Their initial report was in vet immuno and immunopathology. Raylene Woda, that was at K-State, worked on this with them, and they showed that almost all of the patients could develop an augmented IgG response, which is actually then your B cells making antibodies against tumor antigens, and they did that with Western blots. How then do we focus in on a tumor vaccine with this new checkpoint inhibitor? CD200 and 200R, that's receptor, are pretty preserved in cells. And so actually Mike Olin's group, that's just funny that I'm talking about this now, here at Minnesota, I'm doing a locum this week at the University of Minnesota, which is funny. Antonella Borgatti's here, and we've been talking about, this was developed in Minnesota. It's kind of crazy. Anyway, they developed a peptide called CD200-ARL, which targets the receptors of CD200. And they show that it actually can promote the good co-stimulatory molecules like CD80 that I mentioned earlier. It can produce good cytokines by blocking that CD200 pathway. And then it actually can downregulate other immune checkpoints like PD-1, PD-L1, CTLA-4, and some inhibitory things. And so this is a busy drawing, but just to say, it seems to kind of tamper down immune checkpoints and then promotes good inflammation and stimulatory molecules, kind of those 1, 2, 3 things we talked about earlier. They did this here at the University of Minnesota in a canine glioma model where dogs with brain tumors had surgery, and they either got a vaccine from their tumor or a vaccine plus this CD200-ARL. I usually just refer to it as CD200 peptide. It's a lot easier than all the other letters and numbers. This is their Kaplan. Sorry, I'm running out of voice here. The Kaplan here shows blue line is actually dogs. This is from Mike Olin's paper that was in Cancers. Blue line shows dogs that had glioma removal, craniectomy with tumor lysate vaccine and CD200 versus red line is just surgery alone with vaccine. And I think you can see a lot more dogs live longer. Their median survival times in dogs, and this is some of the best data we've seen from canine glioma, was 13 months, and that's again the middle of the road. You can see there's some dogs that lived up to 2 years or more, which is really kind of unheard of with canine glioma. You can see dogs that just got tumor vaccine alone, most of them were dead within 6 months. This was pretty exciting. This actually has now moved this into human glioma clinical trials. They said there was no evidence of treatment-related adverse events. And they go on to say that 5 dogs did develop cerebral leukoencephalopathy, which is actually inflammation of the white matter in your brain. They responded to steroids, so it makes sense that an immune stimulant, if it's a tumor in your brain, would actually then cause you to have inflammation in your brain. Osteosarcoma patients, we don't often think of brain mets, and so I don't think that's going to be a side effect for us, but just something to remember that wherever the tumor cells that are going to get attacked live, you're going to get inflammation. And so just something that we pay attention to. I've done a lit search on CD200 in human osteo, and you can see I'm not making this up. 2 references. There really isn't much that's been researched on this. The first one actually really looks at a long RNA molecule. You can see the group here. If anybody wants to go into this, it's in PubMed and it's for free. But they actually looked at TGF-beta signaling, and they actually said if you could block this long RNA, you might actually interact with the CD200 pathway. And so they didn't actually look at a CD200 inhibitor. They were just looking at CD200 pathway as an immune checkpoint pathway in osteosarcoma. The other one you can see here is the clinical trial now that's in people with glioma. What I last saw was that it's active but not recruiting more patients. I don't have any data on how they've done or where it's at, but I do know that it's closed for more enrollees. And I'm assuming data is being evaluated. I want to say that it closed in early 2024. The NCI's website doesn't have any other updates. I think you can see that's a 2023 snap. I just wanted to make things a little bit more relevant again and talk about checkpoint blockade in human osteo. I think that you guys have heard and seen a lot of these. Keytruda, Opdivo, Yervoy, you can see all of these different drugs here. These are their generic names, have been tried, whether it's PD-1, PD-L1, CTLA-4, LAG-3. There's some other things that are on NK cells like TIM-3. A lot of these checkpoint inhibitors have been tried, and the results I think have really been disappointing in osteosarcoma. They really haven't kind of moved that needle. This paper actually has, I think this is kind of a neat drawing. It's like I don't know, like a buffet, if you will, of all these receptors and all the different immune cells that interact with them. The authors of this paper talked about like, okay, well, if these receptors are there, why can't we turn things on? And I think people have always talked about osteosarcoma being a cold tumor, or really getting that tumor microenvironment to be kind of promoting inflammation, which is anti-tumor. And so do you get adjuvants? Do you get epigenetic modifiers? Can you get cytokines in there? Like, what do you do to really get the immune system to be able to get in and kill osteosarcoma cancer cells? And so I think there's, we have to figure out ways to do that. So I think this study hopefully would be something that helps us figure that out. This clinical trial is in 60 dogs. Our goal is to have 2 arms. It's a bit of a different randomization than I'm used to. We did a 3-to-1 randomization because the USDA really didn't want us to use historical controls alone. And so we used Dr. LeBlanc's study that we worked on with Tim Finn. I was one of the investigators when I was at the University of Missouri. And we have such solid data from those, and we're actually going to screen these dogs and then restage them exactly the same as the COTC22 study. And so when we talked to the USDA, we said, hey, do we really have to enroll more prospective dogs? We know how they do. It's just not great. And so we were able to do kind of a modified part contemporaneous, part historical control control group, and it's 15 plus 157. So in our control group, we'll have 172 dogs, but only 15 are going to be prospective. And then there's 45 dogs in the immunotherapy arm. That immunotherapy arm is autologous vaccine with the primary adjuvant, and then this immune checkpoint inhibitor, the CD200 ARL. Dogs will be screened. They're excluded if they don't have osteo on histopath, or if they have not a viable vaccine to be produced. That's one of the big things here, is we have to get live cells out to be able to make the vaccine for them. One of the big things we're now running into is there's some new monoclonal antibodies for pain in dogs, and those actually could potentially interact with the immune system. And so Librela is the dog that we have now, which is a monoclonal for nerve growth factor in dogs. People are using it a lot, and there's worry about how it would interact with other immunotherapies. And so our hope is that we're not excluding too many dogs that are on new immunomodulators. And of course, then any dog that's had previous osteotherapy is excluded. So we're screening dogs. We've already screened, I think, 3 dogs, enrolled 2. They're randomized either to immuno or chemo. And then our goal is then to monitor disease-free interval or their remission duration. The nice thing for this is, Christina, you mentioned compassionate use. Dogs that have progression on chemotherapy have the ability to get vaccine and immune checkpoint inhibitor for compassionate use. The opposite isn't true. If they have progression on immunotherapy, we know chemotherapy doesn't do much in the macroscopic or metastatic setting. So that's the design for the study. Inclusion and exclusion criteria are here. I'm going to speed along a little quick just because I probably talked too much about the Immunologic Synapse. But this is run through multi-institutional, and the big thing on exclusionary is prior treatment. If they're on, I kind of mentioned those immunomodulatory drugs. A lot of dogs have allergies, and so we also have immunomodulatory drugs for allergic dogs that we have to be careful with. And then one of the big things I'll mention is dogs are often on non-steroidals, kind of like ibuprofen for people for pain. And so really we don't want dogs on NSAIDs because I need the innate immune system to be active. We talked about the relationship between innate immune system and the adaptive. And so we're trying to have dogs not be on NSAIDs around their immunotherapy administration. The schedule looks like this. I'm going to go over it really quickly, but dogs are screened that are standard to how the dogs were screened in the initial standard of care versus rapamycin study. We're then following dogs out after an amputation. They get 3 doses of vaccine and then are monitored every 2 months for chest radiographs to look for metastasis. That schedule looks really similar to the chemotherapy group, same screening, amputation, and then 4 doses of chemotherapy. The treatment packs, this isn't so exciting for the human side, but they actually contain the CD200 peptide and then also autologous vaccine. Just there's a priming dose of CD200 peptide and then a follow-up simultaneous dose with the autologous vaccine, and that's repeated every 3 weeks. So there's a couple injections. The nice thing for this is it's all intradermal, and so we're actually able to give this through the skin. It's not a deep needle. It's not intravenous. It's not intramuscular. It's a really kind of nice little intradermal needle. I'm going to show this really quick just to show you what that looks like in these dogs. I have to mute. These are videos I took when I was out in Maine at work. And so this dog is a German Short-haired Pointer that has osteosarcoma of the right femur. And so you can see his little shave spot on his back. This is where his checkpoint peptide, his CD-term peptide is going to go. This little applicator is called the VAX-ID system. I'm showing there the styletter guard that keeps the needle inside. And so I pulled that out. The checkpoint peptide, the CD-term peptide, is in that syringe. It's attached to the VAX-ID applicator system. It has the tiniest little needle that actually then goes into the skin. You just push your syringe, and it does a really nice little bleb. It kind of reminds, if anybody's had a TB test, you get those little blebs or bumps. So it does a really nice intradermal injection. And this is just a second video of that same thing. It's not that exciting. Difference now, if you'll see the autologous vaccine, this is actually gooey, gross, dead cancer cells from this dog's tumor. So hopefully you can see that red grossness there. And so actually then this goes next to the checkpoint inhibitor, and then we also put it over the draining lymph nodes. So this same system, I actually can now put autologous vaccine into the skin, or kind of through the skin, if you will. I'm putting it next to, you'll probably see those two little blebs. They look like googly eyes on that dog's back. If anybody's squeamish, I'm sorry, but it's not really even a needle. It's almost like the tiniest little prick, if you will. And now you'll see kind of a pinkish red bleb next to those other two blebs. So that's its autologous vaccine with its checkpoint inhibitor, if you will. Then we're also putting this over the draining lymph nodes peripherally. And so this is actually a superficial cervical lymph node that's over kind of where we would have our clavicle or our collarbone. And so you can actually see that little bleb there where I put his vaccine. Goal being is that we have kind of interaction between the vaccine and the checkpoint peptide next to each other. And then I recruit, if you will, immune cells over the draining nodes where all the fun happens. Outcome measures, I kind of already mentioned this. We're monitoring disease-free interval or remission duration. And then adverse events, we have a grading scheme that we use with VCOG to say what our potential side effects are. Immunologic effects, we're thinking of our local, but then potentially systemic. And we already talked about anaphylaxis or other kind of immune. I'm sorry, I'm running out of juice here with my voice. Immune side effects systemically, and then chemotherapy side effects. I think we know about bag side effects with bone marrow suppression, hair loss, and GI upset. Study sites that are open: the University of Illinois, my alma mater, University of Wisconsin, Maine Veterinary Medical Center, where I do part-time work when I'm not on the farm. And then we're also opening up into private practice, which is pretty exciting and newer for me. I've not worked with clinics in private practice before. I've really been in academia most of my life. And so there's Blue Pearl and Thrive clinics around the country. We're trying to get this going in Michigan State, Purdue, and then hopefully Minnesota. It's where the peptide came from originally. And then Ethos, which now has quite a few oncologists involved, is another one. So we're hopefully getting this open around the country, both coasts, the South and the Midwest, and then in Colorado too. So it should be a site around the country. Where to find it? Ardan has a clinical trial link, and then the AVMA website. And then Christina, thank you again for helping us get it on to MIB's active clinical trials. The trials team, Tim Fan is the principal investigator at the University of Illinois. I'm the project manager through Ardan. That I'm in Illinois some of the time, and then I'm in Maine, or I'm in Minnesota. I don't know where I'm at a lot of the time. Adrian is our sponsor representative at Ardan. We have a lead study monitor, Marissa Kroll. She's an oncology nurse. That's fabulous. And then the histopathology reports are run through BDX out in California. Big thank you from me to a lot of people that have trained me and worked with me on a lot of different osteosarcoma projects. Mizzou folks, Tim, Andy Flory, my old colleague and boss at PetDX. I've got Amy and Christina in the group at the National Cancer Institute. And then one of our favorite patients at the University of Missouri with one of my favorite residents. I don't know if she'll listen to this, but Dr. Norquist, I think might be a clinical site as well in Ohio. So that's the spiel about the study and a little bit of the background. And this is the family farm. When cows are coming home, I can take some questions. That's the cow dog on the farm.
Amazing. That picture doesn't even look real. It's so beautiful.
Yeah, it's real.
Thanks
Yeah,
so much. Everyone,
yeah,
feel free to put questions in the Q&A. And thanks. I feel like I got a really good primer on the immune system from the
good,
first part of that. And
good.
so just some questions maybe to kick it off about the vaccine itself. So how long
Yeah,
does it take to manufacture the autologous vaccine? And
yeah.
is it expensive? Like, what does that
That's
mean?
a good, yeah, no, good question. The autologous vaccine. So the big thing with it is the legs or tumors, if you will. But since they're amputated limbs, the entire limb goes to Ardent, and it needs to be alive. So they're processing those the very next day. They're overnight shipped, and then the processing vaccine production is usually probably around 3 days. They have to irradiate them. They have some cell counts to make sure they have viability, and then they add the adjuvant. So there's about a 3 to 4 day window where they're making the vaccine. The vaccine is stable and preserved at their location at -80. And then when it's shipped out, it can be stored at -20. So it is frozen and then thawed before it's given. The vaccine itself takes about 3 to 4 days to produce. Then, of course, then the rest of the tumor goes to the pathologist. Then the decal process has to happen. And so that's one of the things that we're really trying to say, too, is, okay, osteosarcoma, with all of the mineral and the calcium matrix, can we make vaccines that are viable out of all of these dogs? And so that's one of the big hurdles that we're going to try to answer with the USDA is, can we make autologous vaccine for these dogs? And are there some subtypes or locations that really vaccines are hard to make? So I hope that answered your question.
Yeah, kind of. And so what has been the success rate of creating a vaccine from the tumor?
I can't give you an exact stat. And again, I'm not an employee of the company, but I've done this now in several dogs. I can tell you of 2 dogs I've had that we could not make a vaccine. One of those, it just didn't have enough cells. And the other one, I think the cells were growing so fast that they were necrotic before we could even make a vaccine. And so if you have a really high grade tumor that's really turning over quickly, if on arrival all the cells are necrosed and dead because they're just dividing so quickly, necrosis is a big thing to think about in the human pathology of osteosarcoma. We have to have viable cells. And so I have at least 2 examples, many more that it's worked, but 2 where we could not make a vaccine either because not enough cells or they were necrotic. Yeah.
Yeah, gotcha. So,
Yeah.
I was going to actually ask, though, I guess it doesn't make sense in kind of how this trial is, because you're basically amputating and then creating the vaccine from that primary tumor. So I
Correct.
was going to ask if you could first give the vaccine for longer, because I think right now it's like 3. Is it 3
3
different?
doses. Yep.
Yeah, to extend durable response if you're seeing
Yeah.
disease
They,
progression.
They have, I know that Arden has usually saved some extra, if they have it, vaccine that you could think about for boosters. The problem is it is autologous, and so you have to have enough. So anyway, it just really depends on how much tumor is there, and then it's a really kind of a cutoff of number of cells, and you're looking in the millions. There is potential to have boosters, not in this study, but in the future. If you do have a really good response and you're in remission, it would make sense, like other vaccines, to give boosters down the road. And it really depends on how viable and productive the vaccine is, or the tumor is, I should say. Yeah.
Yeah. I mean, would you be able to, because you can manufacture the vaccine so quickly, I mean, 3 to 4 days is pretty quick.
Yeah.
If there were disease progression, could you just, I don't know, do you resect pulmonary mets in canines, and could you just generate another vaccine?
There's actually a study looking at that. It's called the MIMIC study, where they're doing
Right.
metastacectomy.
Yes. Actually, we had Dr. Thompson on.
Yeah.
Yeah.
So Chris is doing that. That would be cool. So I actually think that's a good idea. And that's something that we've talked about is, if a dog, whether or not they were in the study in the first place, if a dog does have progression, you grab that metastatic lesion. In theory, it has a lot less bone in it. And so hopefully it'd be easier to make a vaccine with. So do you make a vaccine out of the metastatic lesion? And then, could you add in an immune modulator with it? Yes, that's exactly stuff that we've talked about. Sure.
Yeah. And so this study is, you're comparing basically kind of standard care, like amputation chemo only, to then just giving the immunotherapy, the checkpoint inhibitor, with a vaccine.
Yeah.
And could you though kind of stagger it? Like you do the
Yeah.
standard care,
Yeah.
infusion chemotherapy, and then add on the immunotherapy after that. So you kind of get rid of, the bulky disease, and then you come in with immunotherapy after.
You've read what I've been doing behind the scenes. So yes, the answer is I think you can either give, and that's where then it becomes, is it concurrent or is it serial? So like, do we give vaccine during the chemotherapy protocol, or do we give chemo and then give vaccine? I don't know the answer to it. I can tell you that clinically I've done concurrent. I've done vaccine commercially, not on a study. They get their chemotherapy and then at selected intervals between their chemo treatments give vaccine. And I think there's a little bit of data on that at Ardan, and I've done it clinically a few times. I don't have enough to tell you that does it work or does it not. So short answer is, I think combining chemo and immuno is attractive. We just have to make sure we have a functional immune system. We're not actually then negating our immune response with chemo. One of the really exciting things that's studied around the country, and especially there's a researcher at Colorado State doing it, is actually, let's say you don't have a surgical lesion, but if you can irradiate a lesion in the body and add immunotherapy in, I mean, that's kind of what we're doing outside, right? We're radiating the cells that we collect to have this immunogenic cell death and then giving it back. If you could combine radiation with like gilvetmab, monoclonal, or with one of these immunomodulators, I think that's pretty exciting. So, yes, but I don't know exactly the best way to do it. So, yeah.
Okay, we actually have a couple questions that came in. So actually, I think this is from Mari. Booster scheduled for trial dogs. So she said with the Yale vaccine, booster shot has been observed to make a big difference for some dogs.
Yeah.
Thanks for that comment. And then also a question about, could you please clarify the immune checkpoint inhibitor schedule?
So there's a priming dose that's given 24 hours before the vaccine, and then there's a second dose given at the same time as the vaccine. And so it's day -1 and day 0, if you will. So like if a dog comes in on Thursdays to the clinic, on Wednesday, you get a priming dose of checkpoint inhibitor, and then Thursday it's vaccine with its checkpoint inhibitor. That's repeated every 3 weeks for 3 doses. And so there's always that priming dose and then a second dose with the vaccine. Yeah.
Okay. And circle back kind of on that other question about, combining immunotherapy with chemo. I thought it was so interesting, the slide you showed with the current trials, the past trials, and the trial that had MTPPE. Canines, where you actually did see there was much longer, progression-free interval. But I thought what you said was so interesting about like not negating the immune response, because in humans right now, the MTPPE is given like with adjuvant chemotherapy. Yeah. And then a little bit after, but it's certainly given when the chemo is actively destroying the immune system. So I think that's kind of interesting learning that perhaps also could translate whatever.
Yeah, we have to really figure out. And that's the thing is, it's and we've had this discussion with other immunotherapy studies, is like, okay, if you give the immunotherapy and then you give the chemo, is the immune system like getting going and then you smack it with chemo, or do you give chemo and then you give? So we have to figure out that interplay of timing, and that's a hard thing that you can theorize all day, but you really need to then do kind of in vivo studies and look at efficacy, but also then look at the immune cells that you're studying and say, what are they doing around the site or in the blood or the lymph nodes? So those correlative markers need to be studied. And I wish I could tell you the ideal timing for all that stuff. And a lot of times that comes from mouse and rat studies. Yeah.
I know, but it would be great to have that learning from canines, because that's very translatable to humans, because we have existing agents, but maybe it's just a matter of how we're sequencing them and timing it. Okay, last question for you, because I know we're a little bit over, but it's been such an interesting presentation.
Thanks.
So again, going back to that, it was so interesting to see that kind of list of trials, which included the trial with MTPPE, which was I think in the early 90s. So I'm kind of curious how that informed the human trial with MTPPE. And then there was also, you mentioned the OSTER2 study that started in canines, which I think phase 2, they just have results now from that in humans. So I wonder if you could just also talk a little bit about this path from canine trial to human trial.
Yeah. So I think there's a lot of times the way things have worked is that dogs are used, but not in like the natural disease model that we do as clinicians, but like as like, like a mouse or a rat study or something else, a zebrafish. And so historically, animals were used as kind of like safety markers to get to people, but not like studying the disease model. And so in veterinary medicine, most of the time what we would do is say, oh, they do this in people, let's try it in animals. And I think that was most the historical model of let's try to recapitulate what they do in people. And yeah, lately, especially with the National Cancer Institute's Comparative Oncology program, and Amy LeBlanc, I think, was on, and I talked about her quite a bit. She's a brilliant researcher and really passionate about this. She's really helped us kind of say, let's try stuff in dogs that we think will work, and then can we use that to inform decisions in people? And so, when I was in vet school, this is a good aside. We were always told cats are not small dogs. So we say, oh, we do this in dogs, we should do this in cats. And so dogs are not small people, so there's a lot of relationship between the two species, but there are things that are very, very different. And so I think our job is to say, hey, these two cancer processes are very similar. We know there's still differences, especially in how the immune systems work in both species. But I think we're really, as a group of veterinary oncologists, we're really trying to say, we want to help our patients, but man, it would really be nice to help people as well. And so I think osteosarcoma, especially Dan Regan, you mentioned Dan is really doing that, especially at the University of Colorado with CSU, Amy at the National Cancer Institute. I mean, there's people all around the country that are trying to say, hey, let's try stuff to help our dog patients out, but then can we use that information to inform human clinical trials and then move the needle and maybe also get stuff available to you guys sooner? Or if something really doesn't work or it makes things worse, you have that information before it goes into a human. Yeah.
Yeah, thank you so much. Thanks for your comment, Mari. She just noted palliative radiation with immunotherapy would be great. And
yeah.
Talk. Thank you so much, Dr. Flesner, for joining us on Osteobytes today and for making it better for osteosarcoma patients, both canine and human. More information on this and all osteobytes can be found on our website at mibagents.org, on our YouTube channel, and at your favorite podcast place. And we're going to be back with osteobytes in a couple of weeks on May 8th. We are going to be joined by Dr. Matteo Trucco from Cleveland Clinic. He also chairs our MIB Scientific Advisory Board, and he is going to be walking us through a guide to the Factor 2025 scientific program. You can find our Osteobytes lineup for the next few months on our website. And if you ever have any ideas for topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Flesner, for spending time with us today. And
You're
thanks
welcome.
to all of you for taking time out of your day to join us. We hope to see you back here soon on Osteobytes. Thanks so much, Dr. Flesner.
Thanks. See you soon.
Thanks.